Peace be upon you all, and welcome to this new episode of Seeing God in Biology. And today we continue our journey with cell membranes. Well, last time we talked about their chemical composition, their potential weapons, and difficulties to have them, like autonomously or spontaneously create themselves in a prebiotic Earth setup. And today we look more with Dr. Fuz Rana into the wonders of design in this magnificent thing called the cell membrane. Faz, how are you doing today?
I'm doing great, Ahmed. How are you?
All is well, and looking forward and anxious to see all the great things you bring with you to us today. So let's maybe go into your presentation.
Sure thing. And just to set the stage again, for those people who make a case for creation for intelligent design, so often the emphasis is on the information molecules in biochemical systems, proteins, DNA, or processes like DNA replication or protein synthesis, the nature of the genetic code. And the one system that is overlooked are cell membranes, and many people just consider these to be these biochemical systems that are haphazard in structure, that are complex, but there's no structure beyond just some initial bilayer structure to the cell membranes. And yet I think overlooking cell membranes is a mistake, because one, these are very important biochemical systems that serve the cell in a number of capacities, but also the more that we understand about cell membrane systems, the more complex and sophisticated and elegant these systems appear to be. And so the last episode, we did quite a bit of a discussion introducing people to the structure and the function of cell membranes. So in this episode, we don't want to retrace that ground. We would just refer people to those previous episodes. But just to give a very quick reset, a very quick orientation, cell membranes are made up of a bilayer. One of the components of that bilayer, one of the major components, are a class of molecules called phospholipids. And there's a number of diverse species of phospholipids that make up collectively the bilayer. And these phospholipid species vary in terms of the length and the degree of saturation of the hydrocarbon chains. They vary in terms of the precise chemical composition of the head group. And these materials, again, interact to form this bilayer structure, which consists of two monolayers of phospholipids that align in a tail-to-tail manner, where the head groups, which are water-soluble, interact with the aqueous phases in the exterior environment or in the cytoplasm, and the hydrocarbon chains, which are water-insoluble, form a hydrophobic bilayer interior. And then embedded in the cell membrane are proteins. Those proteins can be embedded in the membrane, or they can be associated with the surface, either the outer or the inner monolayer surfaces, as peripheral proteins. And it's the different types of proteins that give membranes their different functional characteristics, and are responsible, again, for the different roles that cell membranes play. For example, forming a boundary that separates the interior of the cell from the exterior environment. The proteins that are in cell membranes mediate the transport of molecules in and out of the cell, either actively against concentration gradients or passively with concentration gradients. Some of these proteins serve as receptors, binding materials in the exterior environment, and then triggering signaling pathways that let the cell's inner workings know what's happening in the external environment. Some of these are enzymes that carry out or catalyze reactions that need to take place at the cell surface. Some of them serve as anchors, anchoring the cell membrane to the cytoskeleton in the interior of the cell and to the extracellular materials outside of the cell. So these proteins serve a wide range of functions. And again, I would refer people to the previous episode where we spend quite a bit of time reviewing the structural and functional features of cell membranes. But what we want to do in this episode is talk about how we can make a design argument from cell membranes. And so that's going to be the focus of what we discuss. And so there are a number of ways in which we can make a design argument for cell membranes, in that cell membranes display features or properties that we would recognize as markers or indicators of design. The way I like to make the design argument is following after William Paley in his watchmaker argument, where Paley argued that systems that are made by human designers have certain attributes that reflect the work of a designer. And one of the things that I've noted is in my book, The Cell's Design, is that when we look at biochemical systems, the hallmark features of those systems are identical to the hallmark features of systems designed by human designers. And so some of those features include organization, include fine-tuning, systems that display an organization, systems that display fine-tuning are the types of things that we would produce as human designers. And then also information is something that originates from a mind. And these are three qualities that we see characteristic of cell membranes that in and of themselves could be used to argue that these cell membranes, again, are the work of a mind. And then the last thing I want to bring up is a discussion of what I call the anthropic principle as in terms of its manifestation in biochemical systems and particularly cell membranes. So anyway, we got a little bit of ground to cover. So let's get started. And, again, oftentimes the perspective of cell membranes is that these are just these haphazard, highly complex systems without much rhyme or reason to the complexity of the system, beyond the fact that the proteins embedded in the membrane serve certain capacities or play certain biochemical roles. And yet what we talked about in the last episode is that there actually is a high degree of organization and order to cell membranes. We see that in terms of the phospholipid asymmetry, the existence of annular lipids, the different domains that exist inside cell membranes, which even give rise to these structures known as lipid rafts. And each of these represents a different level, different degree, and different complexity to the organization and the order of cell membranes. And we talked the last episode about the fact that there are a number of different phospholipid species that are found in cell membranes. And these species can differ in terms of the fatty acid composition, but they also differ in terms of the head group composition, where there's a number of different phospholipid species, phosphatidylcholines, phosphatidylethanolamines, phosphatidylglycerols, phosphatidylserines, things called sphingomyelins, which are structurally related to phospholipids. There's cholesterol and other materials, also glycolipids. And one of the things that has been noted is that there is an asymmetry to phospholipid bilayers, that the inner and outer monolayers have different lipid compositions, different phospholipid compositions. And that asymmetry is a precise asymmetry that has to be maintained in order for the bilayer to display its functional properties. That asymmetry also includes the proteins as well, where the inner and the outer monolayers might have different proteins associated with them, and therefore different biochemical activities, different catalytic capabilities. And so that is one level of organization, that there's not a random distribution of phospholipids throughout the membrane or in the inner and outer leaflet, but actually there is a precise distribution of phospholipids that again is connected to their function. And so that is one level of organization that we see. Another level,
it is functional. It is asymmetry that is driven by function that is a marker of design. So, for example, if we look at a surface of a desk and we have a set of letters that are randomly thrown around, then it doesn't capture us as somebody sending us a message. But if we find that letters are put together in an asymmetric way, spelling the word Fuz Rana, then it looks like somebody has written them. And if it is like Fuz Rana and below it it's like Biochemist, then it definitely is not a coincidence to have the words Fuz Rana together. So, the more the asymmetry, it's not just like random distribution of letters. Any letter can be beside any letter, but just the letters of Fuz Rana are together, and then something relevant to them, which is that he is a biochemist, is what captures us, our attention into. If we look at these phospholipids as like letters, each one is like a definite letter, and we find that they're arranged in a certain way that delivers a certain function, then this is what captures our attention as a marker of design. Is this what you are pointing to here? Yeah,
that's precisely it. Yeah, so if we come across an opening in the field and we see just rocks scattered everywhere, we don't think anything of it. But if we see those rocks arranged to form a structure of some sort, and that structure has an obvious utility, then then we would again recognize that this is a marker of design. And so it's that same concept, which is again what you were pointing out, but you were pointing it out in the context of information. But you can even see order that isn't necessarily connected to information that still, I think, reflects design. And that's really the point that I'm making, is that
Absolutely.
That asymmetry is functional, as you're pointing out, aptly so. That reflects design. We also see another level of organization, and this is with the association of phospholipids with proteins that are embedded in the cell membranes. So for example, this diagram on the left shows what are called annular lipids, and these are lipids that have a permanent association with proteins, and they're intimately connected and attached to those proteins through non-covalent interactions, but yet they are intimately associated with those proteins. And people believe that these annular lipids actually serve as a type of lubricant, allowing for that protein to move within the bilayer much more readily, much more easily, which is critical for that protein's function usually. And as a distinction, the middle shows basically non-annular lipids. So you actually have some lipids that are connected to proteins that are functioning like cofactors, where they're not just functioning as a lubricant, but they actually are participating in the structure or maybe even the catalytic activity of that protein. And then you have just simply bulk lipids. So there are some proteins which don't have these kind of direct lipid associations. But again, it's indicating that it's not just simply a random lateral distribution of proteins, of phospholipids in bilayers, but that those phospholipids again are forming, very precise associations with the proteins that are again critical for their function. We also see higher or long range order and organization, where there are these domains that are enriched in certain proteins or enriched in certain phospholipid species that give those domains, very exacting chemical and physical properties that again are oftentimes related to their structural role. And it's a dynamic ordering, because these are domains that are probably changing shape and moving around the bilayer over time because of diffusion, lateral diffusion, and other mechanisms like that. But nevertheless, there is this ordering that exists in cell membranes that is a longer range order. And then last but not least, we talked about the idea of lipid rafts, where these are domains that have precise phospholipid and precise protein compositions that exist as a fixed entity in the membrane that can, again, laterally diffuse, but performs a specific function for the cell membrane. And this is a cartoon that we showed the last time, showing two lipid domains that again are probably understood as lipid rafts that again are dynamically moving around, forming associations, disassociating with one another. So it's again a dynamic ordering or organization. And as we just pointed out, this is a legitimate marker for design. It's not, again, just this happenstance, haphazard membrane composition and membrane bilayer structure. There's a real ordering to it. There's also a fine-tuning to the phospholipid and protein composition of cell membranes. And just here's a few examples. We talked in the last episode about the fact that even that unilamellar bilayer structure is essentially a critical phase. In order to have a unilamellar bilayer, where the bilayer is just a single bilayer phase, requires a precise phospholipid composition, a precise lipid composition. And that we talked about the fact that if that composition deviates ever so slightly from what it is at the particular temperature that the cell membrane finds itself, so in human beings at 30, you deviate, the composition deviates, again, so small, ever so imprecisely from, that particular composition, you lose that bilayer, the unilamellar phase. It collapses into a multilamellar bilayer, which is catastrophic for the cell, which causes essentially a lysis of the cell.
Yeah.
And again, this is again reviewing some material, but we're just putting some of the things we talked about now into context from a design standpoint. As I mentioned the last time, that when you typically dissolve or disperse phospholipids into an aqueous system, they will spontaneously form these multilamellar sheets or these onion-like structures, these multilamellar vesicles. And the only way to get a unilamellar vesicle to form is, again, using very specific types of phospholipids, where you typically have to sonicate the system to break apart the multilamellar structures into these unilamellar vesicles, but that they only remain unilamellar vesicles for a few days before they recollapse into a multilamellar phase. So these are referred to as metastable systems. And yet what we see in cell membranes is, again, a unilamellar structure. And that, again, is formed by a precise lipid composition. And the composition that produces that unilamellar phase depends upon a critical temperature. And so if, for example, our body temperatures deviate to a large degree away from 37 degrees Celsius, one of the implications would be that you would lose that unilamellar structure, or the cell's machinery would have to adjust the lipid composition in such a way as to retain that unilamellar phase. Now, another area where we see fine-tuning has to do with the fatty acid composition as well. And we talked a little bit, I think, in the last episode about the fact that the cell membrane is, in effect, impermeable to at least polar materials. But nonpolar materials will diffuse through the cell membrane. And the rate of diffusion depends actually on the fatty acid composition of the cell membrane. And so the left portion of that diagram shows diffusion again through the cell membrane for nonpolar materials. The central shows what's called a facilitated diffusion. Shows a facilitated diffusion through the cell membrane, where the flow of the material is in accordance with the concentration gradient, where the protein, there's a protein, this is typically going to be for polar materials. Those polar materials are going to make their way through the cell membrane through pores that again, that are moving through the pores in conjunction with the concentration gradient. And both of those mechanisms are referred to as passive transport.
Because it is natural that materials would move from the higher concentration to the lower concentration. This is what it is.
Yes, exactly. And then on the far right is called an active transport, where materials move across membranes. These are again would be polar materials that are moving across membranes, where there is a protein actively transporting them across the membranes. This is an energy requiring an energy intensive process. Now, it turns out that the passive diffusion of nonpolar materials through cell membranes is actually a very important biochemical phenomena. That is how some materials get in and out of cells if they're nonpolar. And this is also a means by which people are looking to do drug delivery as well. That if you have certain drugs that have certain physical properties, they can actually enter into the cell through that kind of diffusion, or hormones, steroid hormones, which can alter gene expression, will actually diffuse through the cell membrane through this kind of mechanism. And so there's been work that's been done trying to understand the factors that influence the passive diffusion of small nonpolar materials through cell membranes. And what they discovered is that this actually depends upon, again, the fatty acid composition, the chain length, the degree of saturation, and the steroid or the sterol content, the cholesterol content. And so in other words, the cell is not only fine-tuning its membrane composition in such a way to ensure that it has a unilamellar structure, but it also is fine-tuning the fatty acid composition so that it allows for the appropriate rate of passive diffusion of small materials through the cell membrane. And so it's like a double fine-tuning, where the cell is actually having to take into account not just a single parameter, but multiple parameters as it's adjusting the lipid composition. And then there's also another type of fine-tuning that we see that has to do with what's called the surface potential. So many of the phospholipids.
So before we leave the previous one, how does the cell actually allow or disallow a material that is not really very needed to pass through? In this case, is it like passive diffusion for everybody, or how does the selectivity come into play?
Yeah, well, for example, you may have some passive diffusion just simply through the bilayer for some organic materials. And so, for example, when you're looking at the cells in the human body, there are going to be certain materials that will enter into the cell that are naturally part of our body's physiology that are going to enter into the cell along the concentration gradient through that passive diffusion through the bilayer. Some of those materials may enter in through pores if they're polar, and then some may be actively transported. You could even have active transporters that are moving nonpolar materials against the concentration gradient. So there's all kinds of different mechanisms at play simultaneously moving materials across the cell membrane. And so, you need to have, again, a fine-tuned,
filter.
Fine tuning to regulate that movement. And so again, it's a double fine tuning that we see.
Yeah.
And in effect, it's a triple fine tuning, because we also have to fine tune surface potential. As I mentioned, the phospholipids, some of them bear a negative charge. Phosphatidylethanolamine and phosphatidylcholine are neutral lipids. They have a negative and a positive region, and so they're Zwitter ions, where the negative and the positive charge are separated in the molecule, but the overall charge on the molecule is neutral. But phosphatidylglycerol and phosphatidylserine bear negative charges, and those negative charges actually mediate the interaction of proteins at the cell surface. So you would have proteins that have surface groups that are positively charged that would interact with the cell membrane through the negatively charged phospholipids, right? And so this is an electrostatic interaction. But when we look at and it turns out that in order for those interactions to take place, you have to have a just right surface potential, which is set up by a just right level of these negatively charged phospholipids at that particular surface. And this factors into as well as into the phospholipid asymmetry that we see. The compositional asymmetry for phospholipids factors into this particular element of fine tuning. Let's see, that's out of that slide is out of I'm not sure. Oh, I know what was going on with that slide. This is this idea of fine tuning of the membrane composition is something that I talked about in my book in The Cell's Design, which we published in 2008, where I was making an argument that the lipid composition of cell membranes is fine tuned. And I revisited that idea in my latest book, Fit for a Purpose. And it's interesting that here's a paper that was published, I think, in 2021 by a researcher from Israel, a guy named Anthony Federman. And he, in this article, is arguing, I don't know if we can see the title of the article very well. I apologize for that.
The fine-tuning of cell membrane lipid bilayers accentuates their compositional complexity. Yeah.
Yes.
It's readable. Yeah.
Yeah. And he's basically arguing that the perception that people have had of the cell membrane is fundamentally incomplete, in that people have not appreciated the fine-tuning, and that he argues that you really should, these two researchers argue that you really should think of cell membranes as a compositionally fine-tuned molecular assembly, right? That they're, again, and he describes three levels of fine-tuning. The phospholipid asymmetry across in the inner and the outer leaflets, the inner and outer monolayers, he argues has to be precisely fine-tuned. The phospholipids, as they interact with the proteins, also has to be compositionally fine-tuned. And you need to get fine-tuning in order to establish the lipid domains or the lipid rafts that are in bilayers. He doesn't even discuss the compositional fine-tuning needed to form unilamellar bilayers in and of themselves. And so this is interesting, because we're now seeing people acknowledging that fine-tuning. And of course, fine-tuning is a marker or an indication of design, right? When you see something that is, again, precisely fine-tuned, and that fine-tuning is connected to some kind of activity that system is carrying out, that again is an indication of design. So that really the organization and fine-tuning are really separate markers for design, but the reason why we consider them evidence for design is a very similar rationale. But to some degree, that organization that we see interplays with the compositional fine tuning. So in other words, to establish that organization requires compositional fine tuning. So even though you could think of them as separate properties, they also interact, or they also interrelate.
Amazing. This method about the electrostatic binding with the proteins with the right charge and the right electrostatic matching between both of them is quite amazing. And does that by any means contribute to a kind of letting in some proteins and not letting in, or just interaction with the proteins in the cell bilayer itself?
I mean, typically the integral proteins have these characteristic hydrophobic domains that will essentially insert into the bilayer structure. But this is something that I don't know. But there is an active, biochemical machinery that is playing a role in the biosynthesis of the cell membranes itself. So there may be proteins that are facilitating the insertion of proteins. Some of it may just simply be, kind of a non-mediated thermodynamic effect. Some of it may actually be facilitated by enzymes. Biosynthetic enzymes. So I don't know precisely how it's probably a combination of mechanisms.
All right, that was very insightful about the electrostatic nature, and yeah.
Okay, so then one other feature of cell membranes that also suggests design is that cell membranes very well may actually harbor information. Now, this is maybe not something that is evident, but where I picked up this possibility was from a paper published, 2001 or something like that, by a researcher named Dorian Lancet. And he proposed something called the lipid world. He's an origin of life researcher where he was presenting another model for the origin of life that would be kind of a companion / competitor to the RNA world, right? Where the RNA world idea is that the very first biochemistry was built around RNA molecules, where these RNA molecules contain information in the nucleotide sequence. And so he would argue that in a lipid world where you would have these liposomes that may be made up of different types of lipid species, that there's actually information in the composition of the liposome. So in other words, depending on that, each compositional permutation represents information. And he argues that information, just like when you have a DNA replicate and that information is propagated to the next generation, that the information in liposomes can be propagated to the next generation as well, where you have a composition for that liposome that likely reflects the availability of different, lipidic materials that would be in the external environment, where there would be some kind of equilibrium established as these materials are hopping on and off of a liposome, so you get a steady state value. And if that liposome grows, it'll eventually reach the point where it becomes unstable and it fissures into two daughter liposomes that would have, in effect, the same composition. And so he argues that this actually may be a way in which you could have information systems that arise out of liposomes that would form as the very first primitive protocells on the early Earth. Now, the merits of this argument aside, I think the key point here is this: that here is an origin of life researcher pointing out that information can also be understood not only in the nucleotide sequences of RNA or DNA or the amino acid sequences of proteins, but you could actually consider the compositional makeup of a system also as reflecting information, where, again, different compositions represent different information states. And so if that's the case, then it's quite possible that cell membranes actually are harboring information in their composition, right? And again, that composition is fine-tuned, and that composition is determining, the passive diffusion of materials through the bilayer. It's determining the way proteins are interacting with the bilayer. And so you could think of that as an informational state, because different compositions are going to impact the fusion rates. They're going to impact domain structures. They're going to impact, the movement of domains relative to one another. They're going to impact protein lipid interactions. And so that the fine tuning that you see, in a sense, you could argue represents a type of information, compositional information, and represents an information state. Now, to my knowledge, nobody has pursued this idea, but it is provocative. And again, if Dorian Lancet ideas have any credibility or any merit, then I think it's legitimate to think of cell membranes as harboring information. And again, information is something we recognize that comes from a mind, right? Your example earlier, that if there's a bunch of letters scattered on my desk, it just looks like somebody dumped a bucket of letters on my desk. But if they're spelling out words, like, welcome to my office, that's obviously, that information is coming from, a mind, right? It's deliberately put in place to communicate a message. And the communication of a message is an activity, it's a function. And so you could think of membranes in that way, or in that capacity. Yeah,
amazing. Except that maybe Lance's idea is probably the information borne by the liposome is going to be mainly influenced by the composition of its environment. But what you have explained about the cell membranes is that the information on the cell membrane is actually controlled by the cell itself. So it has a certain level of persistence of what information it wants to manifest on its membrane. So I guess what we know today about how the bilayer of the cell is carrying patterns that relate to function is a more compelling argument to informational content that is the work of a mind, rather than a proto cell in a prebiotic setting, where the ingredients are primarily influenced by the environment and a certain level of equilibrium with the environment that will cause transfer of information to the next generation. In this case, the transfer of information is taking place because the environment has reached equilibrium, rather than the cell is intentionally moving it around. So I guess even your explanation is more strong in terms of information pairing on a cell membrane, because there seems to be a very tight relation between what is manifested there and what it's doing actually.
Yes, that's an excellent point, because you're right, because in Dorian Lancet's idea of the lipid world, the lipid composition is a steady state, or an equilibrium. The composition of cell membranes is non-equal. It's a non-equilibrium composition.
Yeah, it is because the cell is stubborn.
Right.
Yeah.
Well, I mean, think about this. Like the nucleotide sequence in DNA harbors very precise information, and there are all these repair processes in place to try to correct DNA damage, because you're trying to preserve the fidelity of the information, right? And then even the genetic code is structured in a way that if there is a mistake that's made, that mistake is buffered, right? You don't, it's not manifesting itself. And so the fact that there is this active maintenance of the composition of the bilayer is saying quite a bit about, you know, about the fact that if this is information, it's high fidelity. There's a lot of effort at preserving that information.
Yeah, absolutely. Yeah. So if somebody comes and looks at "welcome to my office" message and scrambles the words "office" composition, but next day we come back and find it "office" again, then something is at play there, of course. Yeah. So the information aspect, I think, is very significant. Maybe even because the cell membrane is not just sitting there. It has a certain level of selecting what goes in and what goes out from the cell, and how to use the environment to the benefit of the cell so that the cell persists. So maybe actually there is some extra nuclear information that is in the cell membrane, or maybe in the cytoplasm even, who knows, that is contributing to what makes the cell remain what it is, in a way.
Yeah, fascinating stuff. Okay, let's see here. We'll keep moving on, and we talked about the. Now, this is something that is an idea that I've been playing around with, and wrote a book about called "Fit for a Purpose," where I explore the possibility that there may be an anthropic principle, or that the anthropic principle manifests itself even in biochemical systems. And so this is a little bit of a nuanced argument, but if we look at the anthropic principle that we're all familiar with, the fine-tuning of the fundamental constants of the universe, you know, many people see the fine-tuning of the constants and say, well, this is evidence for design, and it is. Fine-tuning is indeed evidence for design. But a larger point of the anthropic principle is that there is a fitness to the universe for life. That is that the universe has these fundamental constants that, you know, are essentially endemic in the very nature of the universe itself, and that those values are precisely what is needed for life, and that there's not alternative values that could exist. And so therefore you're saying that the universe is precisely the way that it needs to be for life to exist. And there really are not alternative universes. And therefore, not only is the universe designed, but there is a fitness for purpose. And that fitness for purpose is reflected in the intent of the universe to be a place where life could exist, where life could be possible. And you know, the idea that this anthropic principle extends beyond the laws of physics into chemistry is something that a scientist by the name of Lawrence Henderson advanced, believe it or not, in the early 1900s. And in the next episode we record, where we're going to talk about water, we'll talk about Lawrence Henderson in some detail. But he basically recognized in the 1900s that the nature of water, the nature of organic materials is such that the chemical environment in the universe makes it possible for life to even be possible. That if the chemical environment was any different than what we see, there simply couldn't be life that would exist in the universe at all. So it's a very similar idea, except you're dealing with chemical systems, not with the laws of physics or the constants of physics. And so the idea here is that the chemical properties of molecules are determined by the laws of nature, and these are precisely what you need for life to be possible, and there's not alternatives that could exist. So water has this highly unusual set of properties that are just right for life, and there's no alternatives to water. And so that suggests that there is this anthropic type of property, anthropic coincidence, anthropic principle that we see connected with water. So I took that idea and I asked the question, in the book, does the anthropic principle apply to biochemical systems, right? In other words, are the molecular systems that define life are actually not the product of evolution, of a unguided, historically contingent evolutionary process, but seem to be fundamentally constrained and predetermined by the laws of nature. They are precisely what you need for life to be possible, and there's not alternatives. And when you take these sets of principles and apply them to cell membranes, you see these anthropic coincidences. So for example, phospholipids, right? We talked about the fact that phospholipids are these materials that, again, make up bilayers. Well, when you add phospholipids, at least certain phospholipid species, to water, you disperse them in water, they spontaneously form bilayer structures, right? We talked about that. And it's in part because the head group is water soluble and the tails are insoluble. And so the head groups interact with water, the tails sequester from water, and they form this bilayer structure where the two monolayers interact tail to tail to form that bilayer structure. And then we noted that fatty acids actually form micelles, right?
Yeah.
Even though they have that same amphiphilic structure, they form micelles. So, and we talked in the last episode why fatty acids might temporarily form bilayer structures, but they're not stable. They're not stable structures. They very rapidly will collapse back into a micelle stage. So the question is, why, if both of these are amphiphilic, why do they form very different higher order structures? And the answer to this question was determined, actually, believe it or not, in the 1970s by a biophysicist who developed this concept of a packing parameter, where P is the packing parameter, and P is equal to V, which is the volume occupied by the hydrocarbon chains, divided by the area of the head group, multiplied by the length of the hydrocarbon chain. So this is what he calls the packing parameter. And as it turns out, that the packing parameter has certain values depending on the particular type of lipid, and that gives that lipid a different kind of shape. So, for example, if you have fatty acids, they have a very large head group and a relatively small volume for their hydrocarbon tails, and so they end up forming a cone shape. So if the packing parameters is greater than 1/3, but less than what is about 1/3, you get this cone shape. So then when you then put those molecules together, they naturally will form a spherical structure, right? Now, if you have a packing parameter between 1/3 and 1/2, you'll get a truncated cone, which will still form a micelle, but the micelles will be these tube-like structures. If you have a packing parameter between 1/2 and 1, you get a cone as well, but it's less truncated, and that will now form bilayers. As you begin to put those together, they naturally form bilayer structures.
The diameter will get just larger, the diameter of the superstructure.
Yes, right. And then if you have a true cylindrical structure, which is for a packing parameter of 1, you get a bilayer. And this, by the way, is what the packing parameter is for phospholipids, right? Which is remarkable given that you have, again, a head group structure and these hydrocarbon tails. Isn't it interesting that the volume and the geometries of these components are exactly what you need to get these bilayer structures? And then, like the phosphatidyl ethanolamines that form those inverted micelles, they have a very large tail region relative to the head group. And so this explains, right. And
they
so
flip the other side. Yeah.
Right. So this is why fatty acids are inherently a very poor choice to form primitive cell membranes. Is the molecular geometry is incompatible ultimately. And the only way, well, we'll talk about this in a minute. So you can't really have fatty acids forming, bilayers. There's another possibility. These are called lysophospholipids, where you only have a single chain. Those will form, again, cone-shaped structures. They're going to form micelles. They're not going to form bilayers. And in fact, if you go through all the possible lipidic materials, the only one that is capable of forming bilayers are basically phospholipids, right? Wow. And so phospholipids seem to be these highly unusual molecules that seem to be unique in their capacity to form bilayers. Now, when it comes to that inverted bilayer structure and those phosphatidylethanolamines, they actually serve a very important biological function, because when cell membranes undergo fusion, or they undergo a process called blubbing, which is shown here, you actually have, for a transient, basically reverse micelle phase in portions of that bilayer structure. And so you need to have some phospholipids that actually form non-bilayer inverted micelles, or have that tendency, so that when you have these fusion events, you can have these transient structures that are stable enough to promote the fusion event. So there are these transient non-bilayer phases that are necessary for certain activities involving the cell membrane. Wow. And so when you look at, again, a phospholipid, you have to have basically a glycerol backbone, because you need 3 OH groups, right? And that the glycerol backbone is the smallest organic material that's going to give you, the simplest, it's going to give you 3 OH groups that will serve as attachment sites for the phosphate and the 2 hydrocarbon chains. You need to have a phosphate. You can't have sulfate or arsenate. It's got to be phosphate, because you have to form a diester, right? And you have to retain a negative charge on the phosphate in order for it to have that amphiphilic, that hydrophilic character to form the bilayer. And then you have to have hydrocarbon chains in order to get that interior structure. So if you're going to design a molecule so that it would form a bilayer, you would design a phospholipid. That would be what you would come up with, right? Yeah. So it's not phospholipid bilayers are not an accident of an evolutionary history. They if you're going to have bilayers, if you're going to have a cell where there's a boundary that separates the interior and the exterior, it's got to be a bilayer structure in water, and it's got to be made from phospholipids. That's the only option that you have, right? And so these are, in other words, the laws of nature have predetermined, have already dictated what you have to have in order to have a bilayer structure, in order to have a cell membrane. This is not something that evolution just stumbled upon. This is something that is, predetermined. Yeah. Now, remember, we talked about the fact that fatty acids can form vesicles under some circumstances, and this is why the pH now becomes so important, because when you've got the fatty acid in a ionized state, it forms a very dramatic cone, right? When the carboxylic acid is ionized in a fatty acid, it forms a very sharp cone. When the carboxylic acid is protonated, the cone becomes more like a truncated cone. And so then when you start putting those two together, they start to pack in such a way that you actually get a type of bilayer structure. But if the composition deviates from a 1-to-1 ratio, you're going to wind up collapsing back into a micelle stage.
Yeah.
Does that make sense? Because of the packing becomes unstable. And so this actually now explains, why you can on occasion get vesicles to form from fatty acids, but it also explains why they are unstable, and they always will be unstable, right?
Because geometry wins at the end.
Yes.
Yeah.
Right. And again, it's remarkable to me to think that, again, that not only is the chemical makeup of phospholipids exactly what you would probably design if you're trying to build bilayers, but that in turn, that the volumes of all these different functional groups gives you a packing parameter that is exactly one, right?
Yeah.
I mean, it goes beyond coincidence, right?
It's amazing.
It's a very lucky coincidence that's the case.
If a coincidence at all.
Yeah. And then I'm actually starting to run out of time here, but I'm going to just kind of wrap up very quickly. I mean, another question is, why are membranes so complex? Why not just build membranes out of a single phospholipid species, right? And I think we've already kind of intimated at this, is because these different phospholipid species are doing different things. But as it turns out, that some phospholipids have a very strong bilayer forming capacity, and they have an overall neutral charge. These are the ones that are going to form the bulk phase of the bilayer. Other phospholipids are negatively charged, and those are going to lead to forming highly specific interactions with proteins, particularly electrostatic. With some some phospholipids are going to be responsible for establishing the surface potential of the bilayer. Others are going to serve as metabolic intermediates. And so there's a rationale for the complexity that we see. There's also a rationale for the complexity that we see when it comes to the fatty acid composition, where again, this controls the fluidity, which is critical for establishing domains and lipid rafts. This is critical for establishing the permeability of the bilayers. So the complexity that we see isn't just, again, the outworking of a historically contingent, unguided evolutionary process. It actually, there's a rationale. And when you start talking about a rationale, that suggests that there's probably some constraints with respect to the laws of nature that are dictating the molecular logic that you see, in the compositional makeup of phospholipids. So in other words, you could argue that, the anthropic principle criteria is satisfied, that phospholipids have that they seem to be, their properties seem to be dictated or predetermined by the laws of nature to be exactly what you need for bilayers to form, and hence cell membranes, and that there really are not alternatives to phospholipids. And that suggests that there are these very suspicious coincidences that we see when it comes to cell membranes. So in other words, cell membranes are these highly ordered, highly organized systems where there's organization at the local level and long-range organization and order. There are these highly precisely fine-tuned systems that arguably have informational content in their composition and seem to be the just right systems that you would need for life to be possible. In fact, the only way that you could have life is if you had phospholipids. So when you put it all together, I think it really cell membranes really do look like they've been designed. And when you think about what we talked about in the last episode, that there's a struggle to explain how cell membranes could have emerged through chemical evolution, I think you're really left with only one conclusion, and that is that these systems are really the product of a mind.
Yeah, absolutely amazing. Exactly the right elements are coming together to produce the exact right function with the exact right information, but no path for those elements that are a mandate without an alternative given the laws of nature that we know. There is no path for them to just come together in a prebiotic setup. So the only way would be that they have been brought together by a mind, because life is destined to happen, and the ingredient of a mind intervening or guiding the process to reach to its eventuality is required to leave its fingerprint. It is not an autonomous, fully spontaneous process of nature. Otherwise, we couldn't find the creator. We couldn't find God. We couldn't find any trace or reason for his intentional design to be in the bigger and even in the details of the picture. So I think what you've taught today and in the last episode is that one of the ways to trace the fingerprints of God is to recognize that something like a cell membrane is critical for the existence of life, and the way it is almost a mandate of the laws of nature, but there is no path for this to happen except if a researcher, if a designer, if a scientist has either manufactured it in the lab and put it there,
Yeah.
or the one who created the universe has just left us the evidence right there. So when we are educated enough and we are rational people who don't want to hide behind randomness, we would say, yes, this is a conclusion that laws of nature are engineered for life, but randomness wouldn't have taken us there. The one who has created the laws of nature would be in the best position and the only position to bring together the pieces that he has pre-designed so that life exists. Does that make sense?
Yeah, it makes perfect sense. I couldn't have said it better myself.
I bet you would, but you would have said it better. But anyway, thank you for this great journey with the cell membrane. You say it's just scratching the surface, and it occurs that the cell membrane is the surface. So scratching the surface fits nicely with this episode. So I hope everybody who has seen this has enjoyed it. Maybe plenty of technical details and biochemical acronyms and jargon, but the point is, the laws of nature are made for life, but only the one who has designed them would have known how to put the right components together, and they just don't form on their own. This is the point. So the message for everybody who's listening to this. So I always look at arguments by evolutionists or unguided evolutionists, and they say, hey, look, fatty acids form those blobs, and those blobs can just capture those materials from the environment randomly. And with so many blobs, millions and trillions and hundreds of trillions of blobs forming over millions and billions of years, chances are one of them will be just a protocell that will then be taken by evolution into the path of life. And now the devil is in the details, and I think Fuz was very generous with the details, showing us that it's not that simple. It's not that simple. On the surface, you can just be tricked by these arguments, but when you go to the real details, it's not that simple, and it is real science that takes you to real truth. So thank you very much for teaching and for showing us this great information and those great insights of yours.
Thanks for having me, Ahmed. And again, I hope that people, after watching these two episodes, just have a greater appreciation just for the marvels of the cell membrane.
Yeah, absolutely. So thank you, everybody, and see you again next episode, where we'll be talking about this marvelous molecule of nature, right?
Yeah, water.
Yeah, water. So everything is hydrophilic, hydrophobic this, hydrophilic, hydrophobic that. So this hydro thing is the water that we are all enjoying. So let's get to know more about water from Fuz. And thank you, Fuz, for being here. And thank you, everybody. And see you again soon. And peace be upon you all.